So as I mentioned, I don't study the microbiome, although we will be. You heard about the study that we have in the works with Katie, which we're excited about. But we had a cancellation, and it was about intergenerational biology, and it's something that I study. So it's a topic that I've thought about a little bit, and I think that there are some common themes. And I really want to just make one point in my talk today, and start with this very simple diagram. This is obviously super simplified, right? When we think about why a particular group of people is prone to a disease and another is not, it's a cliche to say that it comes down to genes and environment, right? And that's an idea that goes way back. Clearly, what we mean by genes is becoming a lot more complicated, especially now that we have the microbial genes as part of that story. But the other piece of it that's increasingly becoming complicated is the environment. What exactly do we mean by the environment? And when we think about an organism going out into the world and interacting with the environment, it's things like its diet and physical activity and so on. But it's the environment that they themselves are experiencing. But over the last couple of decades, research has been really underscoring that we need to take a much bigger view when we think about the environment. And I should mention, because we're talking about health disparities, that's what this whole session is about, it's that environmental piece that's crucial, right? Because this is where we have levers, where we can actually change things and improve health and reduce disparities. So this work here, this is work that David Barker published about 20 years ago from the UK, showing that individuals that have lower birth weights have higher rates of heart disease. And he went on, so he passed away a couple years ago, but he really kind of pioneered this idea that adult health traces in part to our earliest experiences, starting even in utero. And so he proposed this model in which, of course, a fetus that's undernourished is going to slow its growth rate as a way to conserve resources. But he proposed that many other things are also changing, right? So lipid metabolism, glucose metabolism, and so on, and that these then persist into later life to influence disease. Now, this model has basically been vindicated at this point, and there are good animal models. I'll just give you a couple quick examples in case you're not familiar with it. So what this is showing is lambs, and these individuals here, their mothers had their diet restricted during pregnancy, and they're measuring cortisol, which is stress hormones, in response to an ACTH challenge. So they're giving the body a stimulant to produce the stress hormone, and clearly they have a different response, right? They have an exaggerated reactivity to that stressor. Here's a similar study. This is guinea pigs, undernourished animals prenatally. Okay, this is insulin, so this is in the fasting state, and this is after a glucose challenge. And you can see that they're overproducing insulin. That is, they're insulin resistant, okay, making them prone to diabetes. And so when we see these simple data like these in humans showing higher diabetes rates in low birth weight, we have a pretty good sense that there's a causal relationship between what's going on prenatally, especially with nutrition, but also hormones and other factors in driving all of this, right? So this model, I think, is pretty well established. What I want to do in the course of my short talk today is try to convince you that we need to take an even bigger view. So when we think about the environment, it's not just even our gestational environment that we need to consider, but also environments going back farther in the past, because we have growing evidence now for pathways linking environmental experiences with biology and health across multiple generations of offspring. A range of potentially independent factors and pathways, epigenetic and otherwise, are likely involved with this. And this is forcing us to rethink what we mean by, quote, the environment. Our health is impacted not only by our present experiences and not only our prenatal experiences and developmental experiences, but also by the experiences of recent ancestors. So these are the themes I'm going to develop. And we're going to do that looking at a couple different types of pathways. The first I'm calling phenotype to phenotype transmission. The second are some of these more contentious, interesting, fascinating examples of germline epigenetic inheritance. Then I'll tell you about some work that we've done that shows that there's plasticity in telomere length, which I think is really interesting. And then finally, I'll have just a few points about the microbiome. Again, this is not my area, so I'll ask a few questions there, maybe plant a few seeds. Okay, so let's look at the first, the phenotype to phenotype transmission. I already showed you the Barker work, right, showing that lower birth weight, et cetera, leads to higher rates of heart disease. And we could fill that in. There's all kinds of things, and in addition to that, we could put inflammation down here. That's another factor that's influenced, right? And so not only is adult health being impacted by one's earliest experiences, But many of these factors, like how much cortisol you have, your stress hormones, glucose levels in the bloodstream, lipid levels in the bloodstream, your blood pressure, all of these in turn impact the gestational environment of the next generation potentially, right? So you have the possibility operating through the phenotype for this kind of effect to perpetuate across multiple generations. And so an example of that is here. So entering pregnancy, if you have hypertension, the risk of giving birth to a lower birth weight baby is quite a bit higher. So not only does lower birth weight predict hypertension, but hypertension then predicts lower birth weight baby in the next generation potentially. We already heard a little bit about the obesity and its kind of intergenerational effects. This is a really, I think, pretty compelling story. So if a mother is diabetic, her glucose levels are high during pregnancy, and her glucose levels determine the gradient of glucose across the placenta and really drive glucose delivery to the fetus, right? So if a mother has high glucose levels, the fetus is going to end up growing larger and having more body fat. They become macrosomic, big at birth, but they also have modified metabolism that makes them prone to becoming obese and diabetic themselves, right? So if they go on to become mothers, they're more likely to be diabetic mothers, and the cycle repeats. Now, of course, you could look at that and say, well, maybe there's just some genetic predisposition, right, that's driving the whole thing that the kid inherits along with the mother. But there are some really, I think, nicely controlled studies that show that there's something about the gestational environment itself that's driving this. So here's a sibling design where they looked at siblings born before and after the mother lost a lot of weight because of going through bariatric surgery, gastric bypass surgery. And you see that the sibling born after was much lighter, had a thinner waist circumference, less body fat, et cetera. You also see that their metabolism is much more normalized, right? So their insulin resistance is down, lipids are down, good cholesterol's up, right? So there's pretty good evidence that there's something about the gestational environment itself that is driving this. All right, so already I think we're seeing that we need to kind of start rethinking this idea of gene-environment interaction. So the classic way that this has been thought of is you inherit your genome at birth, you interact with the environment. Let's say it's an obesogenic environment, right? And you can't really see it here, but that arrow is changing color to reflect the fact that you're changing the phenotype across the life cycle. And then with the next generation, there's a reset, right? Because you're inheriting those genes all over again, you're starting afresh, and then you interact with the environment and gain weight, just like your parents did, right? That's the kind of classic model. But I think when we look at this example of obesity and its transmission, there's another possibility, which is that some of that modified biology here is actually the new starting point here. And again, you can't really see it, but this is light blue, and then it gets darker, and then it gets darker, right? So the idea is that you're not resetting, you're actually doing something cumulative. And so when we think about obesity, which is the true nature of this condition? Is it this kind of generational reset, or is there this kind of amplification that might be happening? I mean, obviously it doesn't have to be one or the other, it could be a little bit of both. And I'll come back to this, because I think that this is actually a potentially relevant model for thinking about the microbiome. All right, so what I'm going to do in the course of this is I'm going to start filling in this chart to kind of talk about some of the environmental pathways that we see operating here. So this is females are open circles, and these are males. And so the Barker hypothesis stuff is showing that the mother's experiences, her environment, influences the offspring. And then when we look at these obesity effects that are multi-generational, you can see that it may even be the grandmother, right, her weight and so on that could carry on to the next generation. We don't know how many generations back that might go. All right, so that's the first example. Now let's look at pathway number 2. So that was phenotype to phenotype, where the only thing that's being transferred are things like hormones and nutrients and so on, right? There's no epigenetic or genetic transmission at all in that case. But here, let's look at germline epigenetic inheritance. And for those of you who need a refresher, when we talk about epigenetics, these are chemical modifications to chromatin that influence which genes can be expressed and which are silenced, right? So over here, you can see that this chromatin is opened up, and so this gene is accessible to transcription factors. And it's the placement of these little chemical tags on the ends of histone proteins that pull the chromatin apart and make it open. Over here, they're away, and this is all clamped down, and that's a silenced gene, right? So these are histone modifications. Over here we have methylation, which is another attachment of a chemical tag that can typically silence gene expression. And then you also have all kinds of forms of non-coding RNA that are turning out to be very important. Okay, so what these are changes in the chemical composition of the chromatin that are modified by the environment potentially, and that persist into later life, but they don't modify the genes themselves, right? So they kind of blur that line between the genome and the environment. But the important thing is that they can be transmitted across multiple generations. So if a pregnant mother experiences stress, 3 generations are exposed. The mother, the fetus, and the future germ cells that are in the fetus that'll become the grandkids, right? So there's 3 generations there. And this also applies to fathers, right? Same sort of idea. When a father's experiences a stressor, he experiences it, but also the sperm that will become offspring and grand offspring in the future. So here's some examples of that from animal model work, where they modified the protein intake of a male rat, right? And then they crossed them with a control, and then they looked at offspring gene expression. And what they found, and they're looking at cholesterol levels. So this is free cholesterol, cholesterol ester. This is in the liver. You can see that there are changes, right, in relation to the control animals. And the punchline here is that the diet eaten by adult males influenced the cholesterol levels in the livers of their offspring, right? So much like Barker was showing with maternal experiences, now we're seeing that there's potential pathways for paternal, the father's diet, the father's experiences to impact the next generation. So that little dot here, that reflects that effect. Here's another example that shows how this can perpetuate beyond a single generation. So this is an experiment in which mice were separated from their mothers at birth as a stressor. Right, so that's the exposed. And then they looked at the sperm of those individuals when they were adults, looked at methylation, okay, epigenetic changes in their sperm. Then they looked for the same methylation changes or the same sites in the neurons of offspring, and then they looked at behavior in the grand offspring in this particular study. And here's what they found. So this is for the cannabinoid receptor, which is related to anxiety and emotion regulation. And you can see that the exposed versus non-exposed have changes in all these different sites along the chromatin around the area of this gene, okay, changes in methylation. Those same changes are present in the neurons of the unexposed offspring. And then they also found that there were behavioral differences, okay, in the generation 3, in the grand offspring. All right, so the punchline here is that early rearing stress in newborn males influence behavior in their grand offspring. So the environment of one generation passes on across 2 generations, operating purely through the patriline. And that's what's reflected here. So environment here, phenotype here, right? All right, so how many generations can these epigenetic modifications persist? And we don't know, but here's a couple studies. Well, this is Anway. This is from Mike Skinner's lab. One of the first studies that really showed these kinds of effects. They treated animals with vinclozolin, which is a fungicide, and it has these endocrine disrupting effects, right? So it's like BPA, that kind of thing. And you see that there are changes in things like testes, cellular structure, and also sperm counts reflecting changes in fertility across at least 4 generations, and they stopped looking at that point. Right, so here, the mother is exposed to vinclozolin while pregnant, and then you get this effect across 4 generations. They also did something similar in females, showing similar kinds of changes in the phenotype across 3 through the matriline. Right? So in theory, some of these things can actually take on a life of their own and really perpetuate for quite some time. All right, the third pathway that I want to show, which is really quite different than either of the others that I've reviewed so far, this actually involves a form of genetic plasticity. I think it's fairly unique. It's a multi-generational effect on telomere length. And this is work that one of our former PhD students at Northwestern, Dan Eisenberg, who's now at UW Seattle, really spearheaded this work. So for those of you who need a quick refresher on telomeres, these are repeating DNA elements sections at the ends of chromosomes. And they're required for mitosis. They're required for DNA replication because the DNA replication machinery needs to be able to sort of overextend beyond the end of the DNA. But it doesn't reach out far enough to actually fully copy the complete sequence of telomeres with each replication. And so you get this progressive shortening in telomere length with age as cells divide. If they get to be critically short, right, too short, then you either get cell death or senescence, cellular senescence, right? And so this is a process that contributes to aging. And so you see that here. Telomere length is long early in life, and then it declines, in later ages. And it matters, okay? So telomere length has been shown to predict mortality. So here you have individuals with longer telomere lengths having higher survival, those with shorter telomere lengths, higher mortality. And importantly, the difference in mortality in this particular case, in this study, was very much related to mortality from infections. And this makes sense because they're measuring telomere lengths in immune cells in blood, and telomere length should influence cell proliferation, right? So immune function should be the kind of system that you would expect to be modified by this, right? To be limited by short telomeres. And we're seeing some evidence from studies like this. Now, so telomeres get shorter with age in all tissues, with an exception of sperm. They actually get longer with age in sperm. And that's because there's high telomerase activity. This is an enzyme that can kind of replenish telomeres. And it doesn't just keep pace with cell replication, it actually increases telomere length with age. And so you see that here. This is in the sperm of young versus older men, longer telomeres in the older men. One of the implications of this then is that children of older fathers tend to inherit longer telomeres. And this has been shown in a number of studies. Well, Dan came up with this really interesting idea. I think it's a pretty cool idea. He proposed that the transmission of longer telomeres to offspring of older fathers could be an example of adaptive plasticity. And the idea is that investing to keep your body going, right, in the long run, to extend your lifespan, could be important to the extent that people tend to reproduce at later ages, right? So this is an evolutionary argument. And the problem with that, though, is birth order effects and other kinds of stochastic influences, right? So if you have the earliest born child to a father and the latest born, maybe there's 20 years of difference between them, they're going to get very different signals about the environment based upon that, right? But they're entering the same environment. And so what he proposed is that because we're talking about telomeres, right? This is DNA, so it's passed on with high fidelity across generations. Perhaps there's a cumulative effect, not just of one generation, but of multiple. And in this situation, this would allow telomere lengths to be increased, slowing aging and enhancing late life function, like immune function and other cell proliferation dependent functions, as average age of reproduction is delayed consistently across multiple generations. So if you have kind of a consistent delay in reproduction, right, across, let's say, 10 generations, then maybe you're going to get kind of a new equilibrium where telomere lengths increase and allow later life function. And he, if you want to read about this, his original article from 2011 really lays it out nicely, and there's a few others that have come out since then. Okay, so let's take a look at this. This is one of the things that he looked at for his PhD a couple years ago, working with the Cebu study that Katie talked about. She already saw some of these slides. So the study began in 1983, enrolled 3,000 plus women, and then their kids are aging along in the study. We got DNA from everybody out here, and this allowed telomere length measurement. So the kids at this age are about 21, 22, and the women are much more variable in age because they're not a cohort. Okay, so hypothesis one, we just wanted to replicate that initial finding. Older paternal age at conception will predict longer telomere length in offspring. And we see that does, in fact, that does hold. So if the father is older at the offspring's conception, you have longer telomere lengths. And if the mother's father is older at the mother's conception, you also see a similar effect size in both of those. Now, how biologically important are these? These base pair changes are kind of hard to interpret, but we compared this to the annual decline in telomere length that you see in the older generation, right? That kind of age-related decline. And it's kind of it's in the ballpark, right? So each year, this says about -12. Sorry, you can't read it. With each year of additional age, you get about a 12 base pair change per year. And here, this is showing that you get about roughly 8 to 9 base pair increase for each year that your father delayed conception. All right, so that confirms what had been shown previously. We then went on to see if this is a multi-generational effect. And again, he was hypothesizing that that would be the case, in part because telomeres are DNA and they have high replication fidelity, but also maybe this would play into that hypothesis that he had about the adaptive role of this effect. And what we found is that it does work. Okay, so this is the father's age at the father's conception is again a positive predictor of grand offspring telomere length, and the mother's father's age at the grand offspring's conception is not. Now, there's evidence from the telomere literature that heritability of telomeres is actually lower through the matriline. So we don't know if this is really a difference, like if this is really limited to the patriline, or if this is an issue of power or some other issue. But it seems to be the case, at least through the patriline, that this holds. So if your father is older at your conception, you have longer telomeres. If your paternal grandfather was older at your father's conception, you have longer telomeres. And if they were both older, you have doubly long telomeres, right? So I think the model holds up pretty well. All right, so what this suggests is that the telomeres here are related to the environment, right, behavior, the age at reproduction, at least in your father and your grandfather. And because this is DNA, it could go back quite a bit farther. And in fact, we have an NSF right now that we're in the middle of that's going to allow us to see how far back that effect goes. And again, we don't really know what's going on with the matriline, right? So we know that the maternal grandfather's age at conception influences the mother's telomere length, but it doesn't seem to make it from the mother down to the kid. We don't know if that's going to hold across populations or if that was just kind of a fluke finding. All right, but the bottom line here is that telomere length may evolve rapidly in response to changes in average age of reproduction in recent male ancestors. All right, so let's look at some concluding points here. We started off with this question: what do we mean by the environment? What exactly is this all about? And again, that's important because if we're thinking about health disparities, the environment is what we can target and what we can change. And I think we have a lot of evidence now that we need to think beyond the environment that as we've conventionally thought of it. It's not just the environment that organism experiences, it's also potentially prior environments. So in the case of the Barker hypothesis, it's the mother's environment. And we have this kind of phenotype to phenotype transmission where obesity begets obesity. We can see this kind of multi-generational effect through the matriline. Going back how many generations, who knows? Probably with a fading effect the farther back you go, but nonetheless, it's probably still there. Then we have this new and more contentious but really interesting evidence for epigenetic germline inheritance, right, where you can get effects through the patriline as well. Some of those effects have been shown to go back multiple generations, so potentially quite interesting. And then we have this telomere effect that I just talked about, which is pretty solid. We've got solid evidence here. Who knows what's going on there? We're going to have to investigate that. But the bottom line is that our health is shaped by the environments experienced by many ancestors. And I'm not necessarily implying that every one of these arrows is important. You can maybe think of every one of those arrows as a hypothesis. But I do think that this literature is pushing us to reconceptualize what we mean by the environment when we think about health disparities. Because it's suggesting that there are complex lagged effects of past environments on our health, and that collectively, we really don't know how collectively important these are. A typical study might look for one effect at a time, right? But they're all going to be operating in parallel and could be quite important in aggregate. All right, so concluding then, what role might the microbiome play in all this? And I'm just going to have a couple quick observations about this because, again, this isn't really my area. Okay, so we already heard some about this. The microbiome is established by early experiences. You have this inoculation from the mother at delivery and via breast milk. And then aspects of that early ecology that's established then persist into later life, potentially. I know there's debates as to how much there's a kind of rewiring of what's going on. And then Laura's work showing that there's actually programming by those early experiences. So even if there is a rewiring of the microbiome and that early effect doesn't persist, there might be persistent biological effects that are secondary to that early experience on physiology, metabolism, and so on. Right? And so here's evidence for, we've already heard about some of this, higher rates of asthma in kids who were born by C-section. Heard about evidence for higher rates of obesity and weight gain, right? Similarly. I think that the microbiome is part of this story. Right? So to the extent that those effects persist into adulthood, right, you have a scenario very similar to some of the phenotype to phenotype transmission ideas that I talked about earlier, where that cycle can then kind of start anew in the next generation, right? As long as you have a situation in which early experiences influence the adult phenotype, and the adult phenotype then influences the starting point of the next generation, then you can get not only a perpetuation of a particular health issue, but potentially the intergenerational amplification of it. All right, and so here's one example of recent microbiome work, where they have 4 generations of animals here, and each one of these columns is a different taxa, is a different microbe, right? And they're giving them controlled diet, and you can see that there's a pretty consistent, there's really not a lot of change in the composition of the gut in this control group. Here they gave them a low fiber diet. And you can see there's a great reduction in diversity, but there's also a shift towards firmicutes, right? These orange are the firmicutes. So you're getting a change in the composition and a reduction in the diversity. The key point that I want to point out here is that the way we've traditionally thought about disease and the way this gene-environment interaction happens is that for this kind of progressive sort of directional change to happen, we would expect that the environment would be changing, that the diet would actually be changing generation to generation and sort of exacerbating the problem, right, with each new generation. The diet is staying the same here. Okay, there's no change in the diet, and yet there's this progressive kind of deterioration in health across multiple generations. And so I think it looks like evidence for this kind of scenario where you've got this kind of amplification, right? And so, I just want to end with this slide where I began. What do we mean by the environment? I think that this example that I just gave is just one illustration of the way that I think the microbiome work can force us to rethink what we mean by that when we think about issues of human health and health disparities. Thanks. Yeah?
Very nice, Chris. This was fantastic. I've thought a lot about this fascinating link between low birth weight and later metabolic disorders. And what really puzzles me is that low birth weight is really 2 different groups of infants. One of them is an infant who completed gestation 40-ish weeks and was just very small for gestational age. But the other cohort within this low birth weight are kids who are growing completely normally up until a point in that pregnancy that the pregnancy ended in a live birth, but that child was, still met the criteria for a low birth weight, whether it be less than 25 grams, whatever. And so in trying to think about mechanisms driving this later, these long-term persistent metabolic effects, I think it's fascinating that both very, very poor nutrition, whether it be from maternal nutrition, whether it be from placental insufficiency that leads to a small for gestational age but term neonate, can have very, very similar effects from an infant who was growing completely normally and just came out of the womb a little bit too soon.
Yeah.
Thoughts on this?
Yeah, no, it's a great question. There's a lot of murkiness in a lot of this. So the first thing to point out is that there's no magic line here where low birth weight is at high risk and higher birth weight is at low risk. There's this continuous dose response. And this little uptake here, so clearly lower birth weight, lower nutrition is one extreme that's driving these relationships, and is sort of the Barker hypothesis that I laid out at the beginning. But then you get into the obesity begets obesity scenario, which is a very different pathway. And I think that's what this uptick is here. But otherwise, you've got a continuous gradation here. So if you're an ounce heavier than the average birth weight, you're still going to have lower hypertension rates and so on later in life, which is kind of a mystery, right? Why is it such a graded response? There's a couple things kind of interwoven in your question. I think one of the sort of distinctions is between fetal growth restriction and gestational timing. generally speaking, you see bigger effects of, that the basic model that Barker laid out and that has been, I think, vindicated is more about growth restriction than timing. There are some long-term effects of gestational timing, but they're not as consistent, and they have more to do with like cortisol exposure typically, so you have those kinds of long-term effects on HPA axis regulation and things that are related to that. But there is a fundamental paradox, and I have not heard anybody provide a satisfactory answer to it, which is that, there really isn't any growth restriction. Is this individual really growth restricted compared to this individual? You've got normal variation. You've got a bell curve of birth weights, and you've got folks with different genetic potentials, right? Who, some of whom are going to be completely happy ending up relatively small, and others who are going to be heavy and also growing very well. Why should that map onto this gradient of cardiovascular disease? I haven't seen a good explanation for it. What I do know is that when you look at the animal models and you restrict diet and so forth, and you kind of push animals this way in the distribution, you get the disease. But I'm not sure if I'm answering your question. But I think that this is mostly about growth restriction, more so than premature delivery. That's the main thing that has been emphasized in this literature. And there is some paradox here because, this individual who's very much over, well above average is going to be worse off than this person here because they're not quite as above average as the other one, And I think it's an interesting observation. Yeah, Bert.
Yeah, I just wanted to follow up on this last question and also connect it up with Barker. On Barker's data, yes, you do see increased rates of heart disease, diabetes among the low birth weight babies, but the point is the bulk of the low birth weight babies don't show these negative consequences later in life, and the question is why. And I think part of what you're raising is there needs to be some more refined picture of what goes on prior to birth to at least separate those out. You might call it a resilience issue. It's something which it always puzzled me that Barker never was willing to address that. And I'm saying that from having had a number of, let me call them close encounters of the fourth kind with Barker. And every time he raised this point, he said, yeah, you're right, somebody should study this. And, we went back and forth for over 10 years on that, but he never did any of it himself.
Yeah, one thing I'll say about that, I'll get your question. In my own, I basically focused on this for my dissertation like 20 years ago or whatever it was. And I tried to get at this a little bit by, for instance, stratifying the sample on the basis of the mother's stature, right? So a small baby is more unusual if the mother's large than if the small baby is born to a short mother. It's crude, but it's one way of starting to get at, is this individual sort of genetically small, or is there something that went wrong in utero in terms of fetal growth? And generally speaking, when you try to correct things in that way, it does strengthen the relationships, supporting the overall idea that, that it is growth restriction that's important. And so we would need to find some ways to try to get at what happened in utero. It's hard. There's ultrasound, there's other ways of doing it, but yeah, so Paul.
Yeah, I was just wondering if you thought about prenatal infection here, because prenatal infection is an important cause of low birth weight, and a lot of these pathogens are persistent. So cytomegalovirus, other herpes viruses, toxoplasma. So you've got prenatal infection potentially lasting for a person's entire lifetime, and they could be increasing inflammatory responses throughout the lifetime because they're persistent, and inflammatory responses are related to cardiovascular disease. I mean, it just seems like there's, again, sort of just like another connection looking in. So have you or other people thought about that aspect?
Yeah,
it seems like
it's a good question. So we have done some work on this, and really this is Tom's area, and Tom, you can jump in a second if you want, but we've done a fair amount on this, but we haven't looked at specific pathogens. What we've looked at is markers and proxies of early microbial exposure, not so much during pregnancy, but in the earliest period of infancy, to see if there are long-range effects on inflammatory regulation and other things, right? So you actually see that lower birth weight babies do have higher CRP in this population. So, I mean, who knows, maybe. But we haven't actually looked at antibody titers, other than EBV, to see, you know, what they might have. EBV,
a lot of these are, you know, well, EBV might be one of them. But one thing that people have been doing is looking at blood samples that have been collected 30 years ago for schizophrenia, for example, and then linking together toxoplasma positivity in those blood samples with development of schizophrenia 20, 30 years later. So you could actually look at that in terms of prenatal infection and directly link it to those individuals that had higher cardiovascular disease risk.
I wish we had blood samples from 1983.
Yeah, they are, but they're there. I mean, you could have...
We don't.
Oh, okay.
Yeah.
You might, I mean, just follow what the others who have done this for, let's say, schizophrenia. Their blood spot analyses, like in California.
Sure, it could be done.
Yeah.
For sure.
So thinking about changes that happen in the microbiome during pregnancy that might imprint, we know that trimester 1 pregnant pregnant women have a relatively normal microbiome, but by trimester 3, it's really changed. I don't know if we have a finer resolution of how things change every time. But thinking about that, what's known about the different gestational windows and some of the epigenetic programming? Do you think that, is there stuff that happens in later development that can still be programmed?
Yeah, I think that, I mean, there's not perfect clarity on this, but there certainly is a lot of evidence for conceptional or even paraconceptional, like really early exposures having particularly big effects. And some of that has to do with how the placenta ends up growing. So if you kind of restrict diet early, the placenta will overgrow as a way to compensate. And, you know, so that'll sort of, you know, there's odd things that are happening with the placenta, basically, that are underlying some of this. Yeah, but there are effects later. And it also, it varies depending upon the system. So, you know, I'm focusing mostly on nutrition here, but there's a lot of evidence for effects of cortisol exposure, because if the mother's cortisol is very high, that crosses the placenta, right? It's a steroid, it goes right through cell membranes. You know, there are enzymes that can deactivate it, but it still gets through, and it has long-term effects. And my sense is that, I don't think we have the kind of critical period for that pegged, but I think that even late in gestation, that can have long-term effects. So to the extent that the microbiome is somehow moderating some of that, it could, you know, I could see that having...
Microbes can actually affect cortisol levels. With the age, they can affect cortisol levels.
Yeah, it seems like they can affect everything, so yeah. So that's an interesting point. Yeah, Jim.
Yeah, I have two questions. Maybe it's not your questions, but the first is in your multi-generational analysis, which is really fascinating for me. The question is whether in studies that kind of vary, you have the states where it goes bad environment, it's bad. But what about intermediate cases where, let's say, I'm just using a crude term, bad, good. In other words, compensation across environments. Is there any kind of way across generations to kind of undo the effects of the finding? I just don't know if there's any experimental evidence or anything like this at all on this kind of intergenerational compensation.
Yeah. Are you talking about the mouse stuff at the very end? Yeah, see, I don't know. And actually, the person who was the first author on that article who was supposed to be speaking in this slot. So she would have been a lot better equipped to answer that. I doubt they've looked at. Do you know the answer to that?
Well, I know that they could rescue the phenotype if they put fiber back in the diet at the end, but only if they also gave a probiotic. So, but these were also mice that were in isolators that weren't getting environmental input of microbes. So you might be able, in that sort of situation, to rescue the phenotype if you had environmental input of some of those microbes. But
and you can track what was going on in the previous generation.
Right.
You know whether the next generation is at risk.
Right.
In fact, we managed to rescue by doing the stool transplant. After third generation, when he put a normal diet, or even one prebiotic, didn't do anything. But when he transplanted the mice and then gave them the normal diet, he managed to rescue, suggesting that there are some taxa lost because of the generational, and that taxa is not going to come back. You have to just replete it, and then he can do it. And the same kind of thing in his human Tanzania study suggested as well, that when you lose a taxa after 2 or 3 generations, it's not easy to replete it with the diet. And if you look at some of the experimental human studies support that. For example, the study done in VA that they give a very carefully done on a high fiber diet to prevent colon polyp. These are obviously 55 and 60-year-old men that didn't do anything. That suggests they didn't do microbiota studies, but I can extrapolate that the taxoid that is lost by the age of 50 is not, with a normal diet cannot be repeated. But you can rescue it, but you have to give the substrate, such as the suit transplant, to be able to rescue it.
You know, and one more thing. The one example that I gave of humans where you have that kind of amplification potentially is the obesity scenario. And the study that I showed, I think, shows pretty clearly that you can reverse it, right? Because there was that gastric bypass. Now, that's a pretty severe intervention. But it shows that if you can normalize women's weight and metabolism through whatever means, it actually will reverse a lot of those effects and stop the cycle. So, yeah, well, I mean, there was huge improvements, you know, in that generation compared to the siblings who were exposed prior. So, it was, yeah, I don't know if you stop it in its tracks, but it's a big reduction in the effect. So, I think we need to eat lunch. Yeah. Okay, let's do it.
Thank you.
Yeah.